Hello students. So this is another video to have an overview of second chapter from second PUC biology, that is sexual reproduction in flowering plants. So in this chapter you will be studying completely about flower and their sexual reproduction. So firstly you'll be studying about the flower, the structure of flower and the complete whorls. So the four whorls, namely calyx, corolla, androecium, and gynoecium. Moving on to, you would be studying about pre-fertilization, the structure and events. So male reproductive structure is the first concept which we'll be studying under pre-fertilization because it is gametogenesis. So under male reproductive structure, stamen, microsporangium, and pollen grains. Stamen diagram can be asked in examination to draw. So this is the stamen diagram and enlarged view of microsporangia. This can also be asked in the examinations. Moving on to the TS of mature ditechous anther. This can also be asked for 5 marks in examination to draw with the labeling. Moving on to microsporangenesis and formation of pollen grains. Please make a note of the definition of microsporangenesis, which can be asked for 1 mark. This is the complete structure which is showing stages of microspore maturing into a pollen grain. Moving on to the sentences or the explanations for a pollen grain. And this is the diagram for a pollen grain in which they would be asked to draw for 2 marks. And again, this is the explanation for pollen grain. Moving on to gynoecium. Under gynoecium, here this is an female reproductive part. So under female reproductive, you will be studying about the carpel, or also called as pistil. So there are 2 types of carpel. One is monocarpellary, one more is multicarpellary. And you will also find another concept, that is syncarpus and apocarpus. Again, monocarpellary and multicarpellary can be asked for 1 mark or as differences. Syncarpus and apocarpus again can be asked for 1 mark for definition or again a differences. Under each carpel, there are 3 parts, that is ovary, style, and stigma. So this would be the overall explanation about the carpel or pistil. Moving on to the ovule, which is present inside. So this is also called as ananthropous ovule. In examination, for 3 marks, this diagram can be asked as draw ananthropous ovule or microsporangium. There are different ways a question can be framed. Make sure you study all these definitions about ovule, because if it is asked for 5 marks, then you are supposed to draw for 3 marks and write the explanations for 2 marks. Moving on to microsporogenesis. This is sporogenesis of mega, that is the female. Make a note of the definition of megasporogenesis. The diagram can be asked as write the stages for megasporogenesis for explanation, or for diagram, draw the stages of megasporogenesis, in which you have to start from this diagram where you can see 1 megaspore mother cell, and then followed by you have to draw this, that is when the microspore mother cell is divided into 2 and become megaspore dyad. Then again division takes place and they become megaspore tetrad. And followed by this A, B, C, D, E. From the microspore mother cell till the embryo sac is a complete diagram for megasporogenesis. But if the diagram is only asked for female gametophyte, then you have to draw from A, B, C, D, E. Or if the question is asked from draw diagram to show the process from or process of division from 2, 4, 7 celled or 2, 4, 8 nucleus, then again you have to start from A and then end till E. This is the diagram you have to follow. And if they ask for mature embryo sac, this E part or the last diagram is the only diagram you are supposed to draw. Moving on to the explanation part. So this is the explanation which is given for the megasporogenesis. Please make a note that it can be also called as 2, 4, 7 celled sporogenesis, or it can also be called as 2, 4, 8 nucleated embryo sac. So this is the diagram which can be asked for. Moving on to the next concept, that is pollination. Make a note of the definition for pollination that can be asked again for 1 mark. Kinds of pollination. This is a definitely asked question. The first kind of pollination is autogamy, followed by 2 types of flower, that is chasmogamous flower and cleistogamous flower. Second type of pollination is geitonogamy. Third type of pollination is xenogamy. These kind of pollinations can be asked, however, as a difference between or mentioning all the types of pollinations, or it can be 1 or 2 as a question. Moving on to agents of pollination. There are abiotic agents and biotic agents. Wind pollination, water pollination, and animal pollination that you'll be studying completely. Wind pollination is simple and clear. Please note down the points. It can be asked again for 2 or 3 marks. Water pollination, again, points are simple. Please make a note of this. And here, as you can see, there's another question which can be asked because there's another type of water pollination which is a little different, that is in Vallisneria. There's another kind of pollination which is taking place. So this can also be asked as a separate question, or in sea grasses. This is again a different type of pollination which takes place. So pollination that is by the help of seagrass. So seagrass pollination can be also asked as a different question. Moving on to animal pollination. Animal pollination can also be asked. The points are very simple. Make a note of it. This is following of animal pollination. Example, for some species which provide safe place for laying eggs are Amorphophilus and Yucca. Then moving on to outbreeding devices to prevent autogamy. This is another question which is frequently asked in the board. So here you have to always mention these 4 points. If this is asked for 3 marks, then 3 points can be mentioned with a little of introduction. But if it is asked for 5 marks, make sure all these 4 points are included with an introduction. So it makes for 5 points. The first outbreeding device is where the pollen and stigma receptors are not synchronized. The second outbreeding device is because of its position. The third outbreeding device is because of self-incompatibility. And fourth is very common because it is unisexual flower. Moving on to pollen pistil interaction. Here it is the recognition of compatible pollen. This also can be asked in the examination, so make a note of this too. The next concept is germination of pollen grains. This concept you'll be working in your lab also, and this can also be asked in your examination, how a pollen grain forms the pollen tube. So this is the diagram you have to draw if a question is asked to draw the pistil showing part of pollen tube growth. This is an LS or lateral section of pistil showing path of pollen tube growth. You can see it in the C part. And the D1 is enlarged view of an egg apparatus showing entry of pollen tube into a synagogue. Then E part is discharge of male gamete into a synagogue and the movement of sperm, that is one into the egg and other into the central cell. So this is also showing the double fertilization. So the next concept is artificial hybridization. Yes, again, this will also be asked for in the examination. So here you have to follow the 3-step, that is bagging technique, emasculation and dusting, and little of explanations. Then moving on to double fertilization. Here you have to explain with the diagram. Double fertilization, you can follow the double fertilization diagram, which is here, and you can write up the explanation part. Moving on to post-fertilization. So till here we had studied about pre-fertilization event and fertilization event. So now we will be moving on to post-fertilization. So post-fertilization major events are development of endosperm, development of embryo, maturation of ovule, that is into seed, maturation of ovary into fruit. So this is completely about endosperm. The sentence part or the explanations also. This can also be asked for 1 or 2 marks. Then moving on to embryo. The explanation is given here. This is the diagram which can be asked as heart-shaped matured embryo. So this is an embryo diagram. Moving on to the embryo, that is dicotyledonous embryo and monocotyledonous embryo. These are the 2 types of embryo. The diagrams also can be asked and the explanation also can be asked. So make a note of the monocotyledonous embryo and dicotyledonous embryo with diagram. Moving on to seed. Seed explanation is given and the structure of the seed has to be explained. So sometimes it will be asked for explanation where you have to write in sentences, and then this is just a diagram to show the seed part, but this will never be asked in the examination, so no need to practice. Advantages of seed, yes, this will be asked. So when advantages of seeds are asked, you are supposed to write to plant and write the points, and to mankind, what are the advantages to mankind, and then write the points too. Moving on to fruits, ovary,